Welcome to our video entitled DNA Replication Made Simple. Very important to understand that DNA is a macromolecule that is made up of nucleotides and arranged in long chains. DNA is a double-stranded molecule, and the two chains will run anti-parallel to each other and will be held together by hydrogen bonds. Some will refer to DNA as a twisted ladder or a double helix, and basically that's pretty much what it looks like. Each of the strands, again, are opposite or anti-parallel to each other, so one runs in one direction while the opposite chain runs in the other direction. There are four bases: adenine, thymine, guanine, and cytosine. The adenine will bond with thymine and the guanine will bond with cytosine. The nucleotide itself is a three-part story. Up here you have your phosphate group, and that is going to be a phosphorus with oxygen molecules around it. So we usually say it's a P or phosphate. Here is your deoxyribose sugar, and here are your bases, which are either A, T, G, or C. When you look at a more detailed deoxyribose sugar, you will see that the carbons are numbered. And in any molecule in chemistry, if there's nothing there, you are to assume, especially at a junction point, that that is a carbon, that is a carbon, that is a carbon, and that is a carbon. And we remember that carbon can bond with up to 4 or make up to 4 bonds. We have carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. Carbon 5 represents the 5' end of the DNA, while carbon 3 represents the 3' end of the DNA. It is also important to note that this is a deoxyribose sugar because you can see at the 2' carbon there is only a hydrogen. If this was a ribose sugar or RNA, there would be an OH there. When we look at how we bond together nucleotides, the numbering comes in handy. You will take the 5' end of, or the 5' carbon of one sugar, and then you will bond that to the 3' carbon on another sugar, forming what is called a phosphodiester bond. In essence, you are building the backbone of the DNA. And once again, the backbone is the sugars and the phosphate. So it's a phosphate, sugar, and so on. The bases are the ladder, not the backbone. When you look at the two strands of the DNA, they are held together by hydrogen bonds. The A, that is bonded to the T, will have 2 hydrogen bonds, while the G to the C will have 3. A more detailed image is here, where you can see the actual bonding. The A to T, that's the first hydrogen bond. This is the second hydrogen bond. In G and C, you will notice there are 3 hydrogen bonds. 1, 2, 3. When we talk about DNA replication, which in essence is taking one double-stranded DNA molecule and turning it into 2 identical copies of a double-stranded molecule, it is important to know that you can only build the new strand of DNA in the 5' to 3' direction, which simply means you can only add to the 3' carbon on the deoxyribose sugar. So in this case, you can see the anti-parallel nature of the DNA molecule, and here's the 3' end up there, and this strand runs that way, while this strand has the 5' up here and is running this way. That is what anti-parallel means. There will always be a 5' next to a 3', so you can see how that goes. Once again, there are your hydrogen bonds in between the two strands. DNA replication is basically DNA being replicated or copied. The original strand, which we know, has hydrogen bonds holding them together, and in order to make a copy, you have to break those hydrogen bonds. Where this breaks, and it's going to be done with an enzyme called helicase, is at the replication fork. And what's going to happen is, as the DNA unwinds or splits, one strand is going to be made continuously and, in essence, follow the unwinding of the DNA. That strand is going to be called the leading strand. The leading strand, once again, is continuous, and as fast as the DNA splits, the leading strand will be copied or created. If there is an A on the parent strand, a T will be brought to the complementary to make the complementary strand. If there is a C, then a G will be brought, and so on. If you look at this diagram, there is your replication fork, and if you see, in this particular case, the leading strand is over here. And the way it is working is, as fast as the DNA can unzip or break apart, this new red or complementary strand is being created. So if there was an A on the parent strand, we have to bring a T. If there is a G on the parent strand, we have to bring a C. And basically what we are making is a copy of the original. This is continuous, this is fast. Notice there are no breaks. When you look at the other side, which we're going to talk about in a few minutes, you can see that there are breaks. And you can see that these red arrows are also, once again, being built in what is the 5' to 3' direction. So that's correct, but it's moving away from the replication fork. So we have this space right here that we have to deal with, and we will talk about that very shortly. So, as fast as the hydrogen bonds are broken, the new nucleotides can be added to the leading strand. Great. Well, the other strand, question mark. We saw those pieces. Let's talk about it. The lagging strand is not and cannot be made continuously. Since these pieces are moving away from the replication fork, it is not a smooth process. You will first need to lay down what's called a primer, which we're going to talk about, and build the small pieces of DNA in, once again, the 5' to 3' direction. These pieces are going to be called Okazaki fragments. Eventually, they will get fused together, but right now they are indeed pieces. Once again, taking a look at the lagging strand, which is over here, you can see this is Okazaki fragment number 1. Let's just say, making this one number 2, and continuing to make several Okazaki fragments. Well, this whole process is going to require several enzymes from start to finish. The first one is called topoisomerase. This has several jobs. First is to help uncoil/unwind the DNA, or to relieve the strain or torque pressure, because of course the DNA loves to be coiled, and to uncoil it does take a certain amount of pressure, and the topoisomerase will do that. Next is going to be DNA helicase, and the way that I'm going to draw that when we draw it is as a little triangle. And basically it acts very much like a wedge would act. And you'll see on the diagram that it is there to break apart the hydrogen bonds that are holding the two strands of DNA together. Once you break apart those hydrogen bonds, now you have these two, in essence, pieces of DNA, or two strands of DNA. And once again, they really want to join back up. They really want to twist back up. To prevent that, we have what are called SSBs, or single-stranded binding proteins. And these are going to act like thumbtacks. Next, now that the pieces are separated and they are not going back together, we need to copy the DNA. Well, you just can't start. It's not that simple. First order of business is you need to come in and lay down a primer. In this case, it's an RNA primer. And the enzyme is RNA primase. This is going to give the DNA a place to begin the copying process. The next enzyme that's going to come in is DNA polymerase III. This enzyme will bring the complementary nucleotide to the growing strand. On the leading strand, this will be continuous. On the lagging strand, you will have pieces, once again, called Okazaki fragments. And each nucleotide is added to the 3' carbon of the deoxyribose sugar. Next, DNA polymerase 1 is going to come in and replace the RNA primer with a piece of DNA. Because once again, you can't have DNA if there's a piece of RNA in there. On the lagging strand, because once again, we did have these things called Okazaki fragments, we're going to have spaces or pieces. And the DNA ligase is going to be the last step that will take those pieces and bind them together, making the lagging strand now complementary and continuous, just like the leading strand. The 2 new strands that will be created are eventually going to coil up and hopefully be 2 identical copies. Assuming there was no mutation or any DNA mistake, the 2 new double-stranded copies are going to be exactly the same as the original. But because each of the 2 strands, one strand being original, one copy, the other strand original and the other copy, DNA and the process of replication is said to be semi-conservative. Each new strand of DNA created will be an original piece and a copy. So let's see it all drawn out in picture form. First piece of business you need to understand is that DNA, once again, is a twisted ladder or a double helix. You can see that the original molecule here I made in red. As you look at the 5' end here, if it's 5' at this end and you follow it all the way to the other end, you will see that there is a 3' end down there. And if it's 3 on this end, it's going to be 5 all the way on this end, and the picture is drawn correctly. We can also see that when we look closer, the bases make up the ladder. Once again, this is your backbone, and your backbone is the phosphates, sugar, phosphate, sugar. And it is the bases that make the ladder. And once again, you have 2 hydrogen bonds here, and you have 3 hydrogen bonds here. And it is the bases that make the steps of the ladder. Again, twisted ladder, bases. If you have an A here, you're going to have a T here. If you have a G here, you're going to have a C here, and so on. So the first step is topoisomerase, and I drew it here as a purple donut, and basically its job is to unwind/take the torque pressure off of the DNA. Once this happens, and again you must remember that it does not undo the hydrogen bonds. There's the hydrogen bonds that are in between the strands or the bases. And once again, if there's an A there, there's a T there. Well, now that we've unwound it, now we got to get it apart. So we bring in our second enzyme. And our second enzyme is helicase. And basically this here is going to break the hydrogen bonds. And what you will notice here also is the strands of DNA are farther apart from each other. And this is going to allow room for the new strand to work, or the new complementary strand to work there. The place where it divides is the replication fork. Well, we got to keep the 2 strands from reannealing or going back together. So the SSBs act as thumbtacks, and they help to keep the strands apart. And that is very important because we have not copied anything yet. Once the strands are apart, they are staying apart. The SSBs have done their job. The RNA primase is going to come and lay down a little piece of RNA. A little piece of RNA there, and a little piece of RNA there. This is going to give the next enzyme, DNA polymerase 3, a place to begin the copying process. DNA polymerase 3 will come in and it will add nucleotides. Once again, if this is the 3', that would make this the 5'. And we are continually adding to the 3'. And basically, as fast as this DNA can be split, or these bonds here can be broken by the helicase, this strand, otherwise known as the leading strand, will copy as fast as it splits. Over here, it looks the same at this point. You've created a piece of DNA. This is called the Okazaki fragment. And once again, let us kind of label this as number 1, just so you can see what's going to happen. As the DNA splits, and as the replication fork moves to the right, which in this case would be this way, the next step is going to be to continue to make the leading strand longer. Okay, that's fine. Well, remember that first Okazaki fragment, number 1? Well, now it's way down there. So a new primer had to be laid down. So DNA polymerase 3 can copy the second Okazaki fragment. And this will continue for a time. In this case, there's our 1, which is way down there. Our 2 has been moved. Now we have a third, or the newest Okazaki fragment here. Notice the leading strand just keeps on growing. Well, now we've done enough, and now we need to figure out what we're going to do. So now we have to get rid of that RNA primer, and we call in DNA polymerase 1. Its job is to take the primer and replace it with DNA. Okay, that's great. And what you'll notice here now is the primer are gone, both on the leading and lagging strand. But we have small spaces still on the lagging strand. Well, the last stop on the tour, and this only happens on the lagging strand, is DNA ligase is going to come in and basically fuse all of the Okazaki fragments. Once that happens, the lagging strand and the leading strand are going to be continuous with no breaks. Notice that you have one red or original strand, one blue, new. Up here, one original, one new. Well, after the Okazaki fragments have been fused, you'll notice once again complementary pairs. If there was an A, we brought a T. If there was a T, we brought an A. And finally, once the DNA is happy and all the mistakes were fixed, it will recoil or retwist, and you will have one original strand and one new strand. These are going to be exact copies, or at least you hope they are, assuming there are no mistakes. And this will happen and allow cells to divide. Well, DNA is huge, so in order to speed this process up, you have to do it at multiple spaces in the DNA at the same time, and we have what are called multiple origins. And here, this would be the leading strand going towards the replication fork, the lagging strand going away. Starting from this midpoint, then we have the leading strand working towards the replication fork and the lagging strand working away. But needless to say, by the time you're done, this speeds up the process. And if you look here, you can see multiple origins, and by the time you end, you started off with a purple and a red double-stranded, started with the multiple origins, and ended up with 2 identical copies. In the world of prokaryotes, the replication is simple. It starts off with a double-layered or double-stranded ring. You start with one origin of replication, you end up with 2 identical copies. In the world of eukaryotes, which are us, that is multiple origins. So we have to replicate quickly, because if not, we would never grow. And finally, I hope you liked this video. In the event that you have any questions, please shoot me an email at psteckel@bmchsd.org or reach out on Remind. Thank you.